Hello, my name is Thomas. I am an MD PhD student, and some of my research focuses on understanding how variants in the gene EBF3 drive the symptoms that are associated with hypotonia, ataxia, and developmental delay syndrome. But today I want to take a step back from that, and I'd like to provide an introduction of some basic principles of genes and genetics that will hopefully help us to better understand the genetic basis for neurodevelopmental disorders as a whole, including HADDS. So before I dive into the science, I want to provide a quick analogy. So I want you to imagine that you are planning a city. And here I have a map of the city of Houston before it was built. So it was essentially the plan that the founders of Houston were hoping to work off of. So you have an idea of the types of buildings that a city requires, right? And you can place those however you like throughout the map, right? And you also know that Houston is particularly flood prone and prone to natural disasters. So you think that it would be wise to not only have one of each of these buildings, but also a second copy of each of these buildings, just in case the first one fails. Now, as you can imagine, designing two copies of each building could get quite expensive, right? You'd have to hire two architects and two engineers, and the cost would kind of pile up. So rather than designing each of these buildings from scratch, you decide to borrow the blueprints from two of your favorite cities, right? And let's just say that you borrow them from one set from Seattle and one set from Austin. So for example, one library will be based on the Austin Public Library, and the second library will be based on the blueprints from the Seattle Public Library. Now, the city of Seattle and the city of Austin aren't going to give you the original blueprints for their buildings, right? Because they still need those, or they may still need those. So instead, what they'd likely do is give you a photocopy of their blueprints for each of these various buildings. And as you can imagine, photocopies don't always turn out perfect, right? Sometimes a corner gets cut off, or a coffee spill may obscure some part of the blueprint. And this may have no effect at all if it's just cutting off a corner that has no detailed instructions on it, or it may have a minor consequence if it obscures a doorknob or something. But if it obscures something major, like an entire door, rendering the building inaccessible, then you can imagine that the effect on the overall building would be a little bit more major. Now, thankfully, you have a second copy of each of these blueprints, and you can rely on those. So for something like a library, a city would be perfectly functional with just one library rather than two. But for something like a grocery store, perhaps the population of your city requires two grocery stores. And if you only have one, there may not be enough food for everyone. And you can imagine that this would significantly impact the functionality of the overall city. So by the end of the talk, I'm going to come back to this analogy and hope to show how it relates to genes and genetics, which really isn't all that different from the blueprints of a city. So first, what is a gene? So our bodies are made up of nearly 50 trillion individual cells. And each of these cells has a nucleus, which houses the exact same chromosomes. And within each cell, there are 23 different chromosomes. And we have two copies of each chromosome, one from our mom and one from our dad. Chromosomes are made up of DNA, which of course is the genetic blueprint of life. And DNA is made up of molecules called nucleotides, of which there are just 4 different types. And DNA is transcribed into mRNA, which then leaves the nucleus and is translated into a functional protein. And a specific stretch of DNA that encodes for a single protein is known as a gene. And the specific sequence of nucleotides within the DNA stretch or within a gene dictates which protein will be produced. And because we have 2 copies of each chromosome, we also have 2 copies of each gene. And we've referred to each copy of a gene as an individual allele. So a quick summary, every cell in our body has the same DNA, which is arranged in 2 chromosomes. We have 2 copies of every chromosome, one from our mom, or the maternal copy, and one from our dad, the paternal copy. And these chromosomes are made up of DNA, and DNA is made up of nucleotides. And the order of these nucleotides provides a blueprint for protein synthesis. And proteins are what are the functional component of the cell. And then a gene is a stretch of DNA that encodes the blueprint for a single protein. So as you can imagine, sometimes there are errors in DNA or variations in DNA that we inherit from our parents. This can be a variation that our parents also have, or it can be a variation that arises in the process of duplicating either our mom's genome or our dad's genome. So they may not have this variation, but we may inherit it because it arose again in the process of duplication. So these variations can have no effect. Perhaps the change in DNA is pretty minor and it doesn't end up affecting the encoded protein, or it can have what's called a deleterious effect, meaning that the change in DNA makes the encoded protein function less effectively or makes it altogether unfunctional. Likewise, there can be variations in entire chromosomes. So as I mentioned, we typically have 2 copies of each chromosome. Sometimes we may inherit an extra copy of a chromosome. We may inherit only 1 copy of a chromosome. We may inherit a partial copy or a partial extra copy of a chromosome, or we may be missing a portion of a chromosome. And as you can imagine, these can have a variety of effects based on which genes are housed in the portion of the chromosome that is either duplicated or missing. So how does this all relate to HADDS and EBF3? Well, in most cases, HADDS arises from small variations in one allele of the gene encoding EBF3. So we still have one functional copy or normal or typical copy of EBF3, and then we have one that is slightly different than normal. And these variations in the gene encoding EBF3 make EBF3 function abnormally in the cell, or make one copy of EBF3 function abnormally within the cell. You also may have heard of a related syndrome called 10q26 deletion or deletion syndrome. EBF3 is on the 10th chromosome or chromosome 10, and each chromosome is named, or it has 2 portions. One arm is called the P arm and the other arm is called the Q arm. And EBF3 is on the Q arm at the 26th band on the 10th chromosome. So 10q26 deletion often involves deletion of EBF3. And as you can imagine, if just EBF3 is deleted, 10q26 deletion syndrome can look very similar to HADDS. However, 10q26 deletion can also, in other instances, involve a greater portion of chromosome 10. And if it involves more genes than just EBF3, this can look very, very different from HADDS. A related disorder is 10q26 duplication syndrome. So this occurs when we inherit an extra copy of 10q26. Right now, this is a pretty well characterized syndrome, but the exact role that EBF3 plays in this is not yet known. We don't know what happens when we inherit an extra copy of just EBF3, and we don't know what contribution an extra copy of EBF3 makes to 10q26 duplication syndrome, because in most cases, 10q26 duplication includes far more genes than just EBF3 alone. So back to our analogy of planning a city. HADDS would be like missing one grocery store, right? So our normal city has 2 grocery stores. HADDS would be like missing one. In most instances, 10q26 deletion syndrome would be like missing one grocery store, one library, and one fire station. Now, again, this can vary based on the extent of the deletion of chromosome 10. 10q26 duplication syndrome would be like having one too many, or an extra copy of grocery stores, libraries, or fire stations. So I hope that this provides a basic overview of some genetic principles that will help you to better understand EBF3 and HADDS. And in subsequent videos, some of my colleagues will provide a little bit more in-depth information about EBF3 specifically and what we know about its contributions to HADDS. So thanks for tuning in.